It is my pleasure to welcome Animesh Ray, who has a long affiliation with the CSRC for many years, and is also being a great contributor to our colloquium. And today he's gonna talk to us about genetics.
Thanks, Jose. Very, very pleased to do this. I wish I were personally there. I always loved this opportunity for going to a campus and talk to you people. Unfortunately, this is not to be this year. So I'm now going to share my screen. Let me hear back if you can see this PowerPoint, which I'm going to bring forward. Can you see the PowerPoint now? And it's on slideshow. Can anybody unmute and give me the feedback whether it's working?
It looks great.
Oh, fantastic.
Thanks,
fantastic. Yeah, thank you. Good to see you. Okay, so the title is Machine Learning Mediated Discovery of Genetic Modifiers of Huntington's Disease, but I will talk a lot about experiments as well. But let me start at a more general place. How do diseases occur? And these days, of course, you are learning a lot and from media as well as papers about COVID-19. The current paradigm is that genes have a lot to do, and I'm not just talking about viral genes, I'm talking about human genes. And genes can affect disease outcome, if not the main cause of the disease, which in the case of COVID-19 is the SARS-CoV-2 virus, which has genes, but the human genes whose products interact with the invading organisms' genes, as well as environment. So we are beginning to accept that human genes in the susceptible person responds to the disease causation by modifying the disease syndrome and the modality. And there could be two different kinds of gene variants that can influence the outcome of a disease. One is susceptibility variant, which could be SNPs, DNA nucleotide sequences that are generally prevalent in the human population, and they're not sufficiently rare to call them mutations. And the second are Mendelian variants, which are rare genetic variants, which have distinct phenotypic outcome. And of course, the environment. And I put infectious agents, including viruses, within the environment. And they may have genes, or physical, chemical, hazardous factors, etc. And the understanding now is that there is this triad in reality and network of interactions in which genes interact with themselves and genes interact with the invading organisms if the disease is because of invading organisms and their genes products. And they all interact with the environment, and the environment in turn affects the genes. So it's really a network which is intricately interconnected, and hopefully someday in a remote future, we would begin to understand the mechanisms behind many of these in more detail. Now we are beginning to accept that for most common diseases, genes play a very important role. And I'm pretty confident that in the years to come, we will, if the gene is expressed in all different kinds of cells, you take any cell type and you'll see gene expression, strong gene expression and protein expression. Why is it that only the brain cells degenerate because of this particular mutation that increases the number of CAG trinucleotide? And this is not uncommon because all of those 30 different kinds of cerebellar ataxias that I talked about, they all show trinucleotide repeats, not in this gene, but in other genes. So there seems to be a common mechanism. There may be a common mechanism in which repeated DNA, which codes for a particular amino acid, in this particular specific case, either glutamine most commonly, or sometimes proline, a large number of such repeats causes this neurodegenerative type of disease, but it's cell-specific within the brain. So people were very interested to find out what happens in the chemistry of the disease. And the more they look, the more they find that all kinds of things undergo abnormality. For example, synaptic transmission goes defective with time. And many of these have been gleaned in terms of model organisms like Drosophila, for example, or mouse, or C. elegans, and I'll describe the Drosophila part and the mouse part later. They find that certain neural cells in which the mutant form of the protein is expressed, they may degenerate. May not in certain model organisms, but in some model organisms they do. And they degenerate because of a large number of molecular processes. One of them is inhibition of the synaptic transmission, inability to produce brain-derived neurotrophic factor, which is a growth factor for neurons. A process in which the inflammatory cells are hypersensitized, have to do with this gene product called NF-kappa B, which normally fights infections and also is related to inflammation. In fact, NF-kappa B has been implicated in severe reactions to the COVID-19 patient cases also. So inflammation in the brain through these NF-kappa B mediated pathway, which is turned on in the glial cells, which are cells that are not nerves but are essential for brain function, probably interact with the nerve cells to feed them to all kinds of things. They become predatory. But within the nerve cells themselves, all kinds of other things go on, including interference with the synaptic transmission. For example, the mutant protein is associated with a proteolysis of the protein, a fragmentation of the protein, and a fragment goes inside the nucleus, which tends to interfere with gene expression. It appears that the protein itself, the mutant protein, that is the expanded polyglutamine protein, precipitates and abnormally folds, and as a result, the proteolysis process, the processes inside the cell that takes care of unfolded proteins that misfold and degrade them, these processes may become saturated or somehow insensitized or maybe hypersensitized, and as a result, they triggered the mitochondria, the energy powerhouse of the cell, to trigger so-called programmed cell death, and as a result, the neurons die. And early results, by early I mean about 20 years old results, seem to indicate that this may be a chief mechanism by which neuronal cell death occurs. With so many things, we feel like, the blind men of Hindustan, who were all looking at different places of an elephant, but failing to understand what that animal is. So let's go a little bit deeper into this. So we said that the CAG repeats expand in the diseased individuals. The expansion is classically thought and shown to be due to expansion in the germline of the previous generation. In other words, mainly the sperm cells, but also the egg cells of the father and the mother, respectively. And then the would-be patient inherits those increased length of the CAG repeat into the embryonic cells. And as you know, CAG encodes glutamine, and it is in frame of the first exon. So where normally one encodes a rung of about 17 to 25 glutamines in that N-terminal fragment within the exon 1, in the patients that inherit this mutation, the length is increased to 36 or more. Repeats above 40 are 100% penetrant, meaning that if somebody has one gene out of two genes that one gets from the mother and the father, or the father, one will develop the disease, 100% penetrant, that's what it means. However, when will this person develop the disease depends on the number of repeats above 37. And the longer the repeat, the shorter is the age of onset. In other words, by shorter, I mean the earlier is the age of onset. And on average, Huntington's disease is manifested from about 35 to 40 years of age to about 50 years of age. In other words, people are struck down in their prime period of activity. But some people are affected very early on. The youngest affected was a 4 years old who had a very long repeat, more than 200 repeats of the CAG. If you take the mutant protein, mutant meaning this polyglutamine expanded protein, and compare it with the normal length protein with about 17 to 25 glutamines, it seems that the normal human protein, when expressed in either yeast or fly eye cells, Drosophila eye cells, or in the neurons of the worm Sinorhabditis elegans or muscle cells, and depending on what allele you choose in some mouse brain cell as well, but not in every case, the mutant form is toxic to the cells. It's clearly demonstrable in human cells in culture, in yeast cells and Drosophila eye cells and worm neurons, but it is not clearly demonstrable in mouse. And although you do have some aspects of the disease, unless the total length of the repeat is above 200, the brain cells in the mouse do not degenerate as they do in the human cells. But they do show signs of other aspects of Huntington's disease. Not neurodegeneration, but memory loss, inability to learn after a certain age, and very interestingly, onset of diabetes, early onset of diabetes, and early death. That's what happens in mice. In rat brain, certain cells, the protein is toxic. So it looks like that particular aspect is common to many cells. The normal protein is not toxic, but the abnormal protein is toxic in many cells where one looks at. And yet, the toxicity is not evident in the rest of the human cells. You can take most types of human cells and express the mutant human protein, and you will see no difference. So clinicians have come up with a so-called CAP score, which is derived by the age of onset times the number of CAG repeats minus 33.66 in an empirical equation that gives them a score by which they can predict how badly people are going to be affected at what age when a patient comes through the door. And typically, the CAP score, normalized CAP score is 100, and that corresponds to about age 45, by which a typical adult who has at least one copy of this mutant form of the gene shows the phenotype of the disease. And what kind of phenotype? Well, there are many different kinds. I mean, you can't do biochemical tests with the brain. These days you can do functional MRI and things like that, but it's not always done because it's so expensive and difficult to do when a patient walks through the door to a neurologist's office. But they measure functional abilities by various psychometric means, and this is an inferential curve which shows that it probably declines slowly first and then rapidly later with age. And around this time, you can diagnose it clinically because the effect is sufficient. And the functional abilities are of different kinds. Many of them are motor processes, that is having to do with movement. The patient develops tremor and weakness in the lower extremity and shaking of the hands. And later on, it leads to paralysis, chorea, which means uncontrolled movement of the limbs, especially the forelimbs. And associated cognitive impairment, inability to learn, forget easily, dementia, and things like that. They increase with time. So there are two models as to how this disease occurs, based on the fact that the disease onset is sort of slow and is inversely proportional to the length of the poly-CAG repeat, and it rapidly becomes progressive and very intense. So one hypothesis, molecular hypothesis, is that it is the protein, the toxic protein accumulation is the causative factor, and the more the protein accumulates in certain brain cells, the higher is the more is the rate of progression. And therefore, if the starting copy number of the repeat is high, then you start at a high enough toxicity and you go very quickly downhill from there. And that's why the 4-year-old child became sick so fast, because the child had a very long repeat. And implicit in this is the postulate that the polyQ length is constant once a person is born. The repeat length doesn't change in the cells, somatic cells, or the cells of the brain. And then there is a less popular model. In fact, nobody believed this, I would say 6 years ago, but now people are beginning to wake up to it. And that's the somatic instability model. And that is a person who gets, let's say, 40 repeats, the CAG repeat in the DNA is unstable somatically in the embryo and in perhaps even, the fetal brain, as well as in early childhood, and perhaps even in older patients. And this CAG stretch, because of the vagaries of DNA replication and DNA repair replication and recombination, can expand and contract as well. And when a vastly expanded clone of cells happen to reside within a critical neuronal pocket in a patient's brain, you get the disease in a very severe form. So the severity of the disease, although it is determined by the initial length of the repeat, the repeats are unstable, and the longer the repeat, the more unstable they are, and higher is the probability of a somatic clone of cells to, by chance, by recombination, get a even longer repeat. But of course, you can say the opposite is true. In other words, some clones of cells will get an even shorter repeats. And indeed, that's the case. Not exactly very, very definitively determined, but there are people whose siblings, identical siblings, have inherited So these are, I'm sorry, these are, yes, these are identical twins. So they have inherited identical original size, the germline size of the repeat, actually exhibit very different disease onset modalities. Some are very early, some are much delayed. Of course, one can argue that this is because of the environment, and that is the complicating factor, because people have not looked yet, because these are rare enough, they haven't looked yet at the somatic clones very carefully, the size of the DNA sequence of the somatic clones. This is a 2017 paper in the Scientific Report of Nature, which suggests that incomplete splicing of this gene, remember that it has 67 exons, may have something to do with the disease modality. There are other additional factors, genetic factors, that influence the splicing and therefore causes heterogeneous RNA lengths because of differential splicing. And also the repeats themselves expand, and together they may account for all the variabilities that you see in the disease modalities. And this is the data published many years ago, which shows the variation of the age of onset that is clinically detectable by neurologists. So the earlier, the more severe form of the disease, that's what one assumes, and the CAG repeat length. The CAG repeat length measurement is kind of problematic, and these are measured on the basis of blood cells, not obviously brain cells, because one doesn't want to do that. So you see that there is a large scatter, and regression analysis shows that about 33% of the variations in age of onset, you cannot explain it on the basis of the CAG repeat lengths alone. Noting, however, that the CAG repeat length is in the blood cells, not in the brain cells. So we really don't know what the CAG repeat length heterogeneity is or homogeneity is in the brain cells, and especially the brain cells that degenerate. But suffice it to say that there's a large variation which cannot be explained by just the repeat alone. And when we look at the molecular mechanisms, and I will not go through this, this is only to impress upon you that it's complicated, all kinds of things happen. You can easily imagine that genetic variations, the allelic variations that I showed in the first slide, that occur in the genes encoding many of these proteins that seem to be interacting with the mutant Huntington's protein, can subtly influence the interactions of the mutant protein with these other components, because many components are interacting, and therefore the allelic variations could influence the disease outcome if the allelic variations determine the physicochemical interactions themselves, the kinetics and equilibrium constants of the interactions. What this tells us is that many other genes would have alleles that should modify the Huntington's disease outcome, not just the CAG repeat variation. And if true, we ought to be able to see the signatures of such genetic modifiers within the allele structures of human populations. So let's go back to very basic genetics and think about what kind of genetic modifiers there might be. So you can think of a molecular mechanism in which the mutant Huntington's disease protein produced a toxic effect, its accumulation produces a toxic effect by whatever means. Then there could be a modifier gene X whose mutation suppresses the toxic effect of the mutant Huntington gene. It's very nice to have such genes because these may be therapeutic candidates, because it's far easier to knock out the function of a protein with a small molecule, small chemical, than to increase the function. So if a gene mutations are generally loss of function type of changes in a protein. If a mutation knocks out the toxic effect of the mutant protein, then so can you by finding a chemical that knocks out the function of the modifier gene's product. But then there could be genes that enhance the Huntington's disease. Loss of function mutations can enhance the Huntington's disease modalities. And so there may be many ways by which it can do that, and I wouldn't go into it. But those genes are not very useful, although they are very useful for understanding how Huntington's disease occurs. But they're not immediately useful to give us a therapeutic candidate molecule. However, almost always if there is a modifier gene that enhances Huntington's disease, then it might have a modifier gene that suppresses or represses its effect, inhibits its effect. That gene can be a therapeutic candidate. And in fact, mechanistically, that gene Z is nothing but the equivalent of gene X. In other words, this line is simply a genetic formalism, using Occam's razor. In effect, the modifier gene X may be a negative regulator of, or an inhibitor of modifier gene Y or its product, and it itself may be an inhibitor or a negative regulator of the toxicity of mutant HTT. So you can play this genetic game in your mind and think about how such things can exist. The question is how to get them. So there are two ways by which you can get them. One is to look in a genome-wide fashion, because you can sequence thousands of genes nowadays and look at all the variants. You can look at thousands, in fact, hundreds and hundreds of thousands of people for genetic variants and find compartmentalized people who are more susceptible to Huntington's disease or less susceptible to Huntington's disease, and track down some genetic loci with some alleles that are hyper-represented in those compartments. And this has been done, not very successfully. And I will tell you a little bit about that later. The other way is completely intellectual rather than the brute force of genome-wide searching. And you can begin to think about how anything works in biology, and how we find out how anything works in biology. And the allegory that I use is my LinkedIn network here as of a few years ago, that I am who my associates and neighbors are. Similarly, a protein's function is encapsulated by with whom such a protein, let's say this guy, interacts. So if we know the function of this protein and that protein, then we can probably infer the function of that protein, and similarly here. So direct interaction between or among proteins betray related functions, just as direct communication with you and some of your associates can betray related interests. So started Sonali in my lab several years ago, and her starting point was to put together the databases of such interactions of genes and proteins, and to some extent genes and proteins that we know in the Huntington's disease affected organisms, either normally in mouse or abnormally in experimental or other experimental organisms which don't have sophisticated brains, such as yeast. So she began with a data set of a genome-wide suppressor screen in yeast published in 2013, which gave her 317 open reading frames that, if mutated, loss of function mutations in these 317 open reading frames or genes, that suppressed the toxicity of human Huntington's disease protein. In other words, proteins normally expressing the normal human gene for Huntington's protein, perfectly healthy, but expressing the mutant gene dies, and then these authors mutated other genes in the yeast genome to find out, as a result of these mutations, which of these mutations the yeast cell survives. So in other words, the gene X in the previous formalism. So they found 317 of them. Then there was another genome-wide loss of function screen. This is based on sheer mutagenesis experiments, even older, 2005, and got 28 of them that suppressed the mutant function. Then another one in which there was 52, and these were done with slightly different alleles of the human protein, toxic protein. This used 103 polyglutamine repeat. This used 53 polyglutamine repeat. So this is milder in yeast. So they got additional 52, which are different from these 317, by the way, or these 28. We several years ago looked to see whether, for a totally different reason, whether genes that encode ribosomal proteins and ribosomal RNA can suppress the toxic effects of the Huntington's gene. And we found 41 of them have some effect. So we put these into the data set. And in addition, there are models in yeast. Oh my goodness, I'm badly out of time. Okay, so I'm going to rush. In mouse, they did similar experiments in which the human toxic protein was expressed, and then they found which proteins physically interact with the human toxic protein, which protein in a mouse brain. So using mass spectrometry, they identified these proteins. So what Sonali did is that use this kind of formalism. Let's say you have genes A, B, C, D in yeast, which all interact with the protein A. Then do their homologs exist in mouse? And sometimes they do, sometimes they don't. And sometimes they have additional ones. And then from there, do they have homologs in humans? Using this kind of gene and protein-based homology studies, she put together a large database of human orthologs that have basis in either yeast or mouse interaction. By interaction, I mean either suppression or elevation or enhancement of Huntington's disease gene, and put them together in this hybrid virtual database. Remember that this is not a reality, it is only an expected reality, because these are based on homology models. We call this Huntington's disease integrome. And then we want to see if there are patterns in it that explains possible genetic modification of the Huntington's disease outcome. 16% of the proteins in the human interactome, protein-protein interactome, actually happens to interact with a mutant Huntington's disease protein. And this interactome was then analyzed by two different means. Are there hidden patterns? So one is the unsupervised machine learning, and generally, it's analogous to clustering, some sort of clustering. And usual clustering methods used in biology uses hierarchical clustering or topological based clustering that assume non-overlapping clusters. That is, members of two clusters do not belong to more than members of two clusters are exclusive. In other words, one protein does not belong to two different proteins, protein clusters or two different gene clusters. But that's not optimal, because we know that many proteins have more than one function. In fact, 207 out of 1628 proteins that were hand curated in yeast belong to complexes, different complexes. So that kind of clustering is non-optimal. So we used a kind of clustering in which two genes, G1 and G2, were put into one cluster if they had some overlapping function. And they may belong to two different, each of these proteins may belong to two different clusters because of other aspects, other properties. And what we did was start with single nodes, grow networks around them, networks meaning interaction networks, and find overlapping clusters by a function known as cohesiveness function, which is basically the ratio of the weights of within class edges times the number of vertices, that is the nodes that are within that class, divided by the total number of such edges. And she found many clusters, but the p-value, the significance compared to a random model, sharply deteriorated beyond 10, cluster 10. And the first few clusters we took, and then we looked at the robustness by injecting random noise into it or by deleting nodes randomly, and we found that the first few clusters were pretty robust to that, using the Jaccard index as the measure of robustness. And these are the clusters, 1, 2, 3. That's, oh well, I didn't show the 3. What are they? The top cluster, which I don't show here, had to do with protein folding as anticipated. And if we hadn't found them, the method would have been spurious. So we found them, that's great. But the second and the third cluster and subsequent 5 clusters were all enriched for DNA damage repair function. And that was surprising, because we had been talking about things mostly about protein folding and generally not about DNA damage repair. And so here. So we went back to yeast genetics using original Susan Lindquist's model, in which the physical interactors of wild type and mutant HTT and the genetic suppressors or enhancers of the wild type and mutant human HTT were looked for using the awesome power of yeast genetics. And so the goal is to generate the hypothesis for future testing and validation. So we did that, and I'm going to skip all of this, and found that indeed there are genes that are required to deal with chromosome condensation and chromosome separation, and genes having to do with that, they either enhance or suppress the toxic effects of Huntington's disease protein. And we think that that's happening by 2 mechanisms, either the bypass that prevents DNA entanglement or a bypass that facilitates DNA disentanglement. And these are some of the data, and I will not go into the data. These are some of the pathways that we mapped, in which the chromosome condensation, decondensation, and chromosome dynamics complexes of the yeast protein interacts very interestingly with some of the genes that are already been worked out in human cells and in model organisms that have something to do with neurotransmitter release and having to do with this kynurenine metabolism, which is a part of the tryptophan and tyrosine metabolism. They related using genetic means the 2 modalities we were able to relate. And this is a pretty picture of what that particular relationship is, and not very worthwhile looking at it too much unless you're very deeply interested in this. But one thing to remember is that the DNA repair recombination and chromosome dynamics genes are intimately connected to kynurenine metabolism, which involves mitochondrial function and neurotransmission. So next few minutes, we will talk about supervised machine learning. And the hypothesis behind this supervised machine learning model that we used very successfully, again, was the idea that protein-protein interaction would have necessarily something to do with the protein sequence motifs and the protein structural domains that enable the specific protein-protein contacts, structural determinants. And the second hypothesis is that, and this is not obvious, is that how the whole network of proteins interact with one another, the abstract topological properties associated with each member as a membership of the entire network may teach us something about the property by which these genetic modifiers might interact with the mutant Huntington's disease protein. So with these two hypotheses in mind, Sonali trained a large number of machine learning algorithms, supervised machine learning algorithms. And to do that, you need a feature vector that have something to do with the features of the structural motifs and domains and features of the topological parameters of the protein-protein interaction network. And I'm going to skip that and talk about these. So motifs, structural features from uniprot, domains, which are combinations of motifs that fold together and are found discretely within the proteome of various organisms. These are domains, so domains are bigger than motifs. And then graphical properties of the proteins. For example, average shortest path length, betweenness centrality, closeness centrality, clustering coefficient, degree, eccentricity, all of these. We don't know which one is going to work. So we took all of these. And we also, because these are very large number of dimensions, we found that we could gain by doing principal components of all of these topological motifs in different combinations, and then using the first, second, third, fourth, etc. principal components as features themselves. So in other words, we reduce the dimensionality to make the algorithms work and evaluated whether the algorithms are working better or not worse. So the input data format for the classifier is you have protein number 1, 2, 3, 4, etc. We interact with other proteins, and either they interact with a mutant Huntington's disease protein, or they interact with a normal Huntington's disease protein, or they interact with both. And so they have motif number 1, do they have or not, 1 or 0? Motif number 2, do they have 1 or 0, etc. And then a series of topological values for each of these proteins in that network. A very simple idea. And sometimes some of these are compressed into principal components. And it doesn't work for most of them. We tried all kinds of things, including Lasso regression, random forest, etc., etc. What worked is gradient boosting model, which makes many decision trees and iterates on the residuals, trying to impute a function and minimize the residual error term using a gradient that gets it better and better. And indeed, this worked. And in the best case, we had, with tenfold cross-validation, we had the machine working at an AUC of 85%. That's great. The best cases were when we applied certain filters with respect to the variables, feature variables that we used, using certain criteria in which we used the information value of the motifs and the domain variables to pare down on certain motifs and certain domains. The machine was more predictable. And this shows the various predictions of the various combinations of motifs and variables. And so some of them don't work much and others work. And in terms of the ranking of the various variables, it looks like degree of the protein, number of direct interactors it has, is a pretty strong predictor. Principal component 4, who knows what that is is about equally good. Average shortest path length, which is not a local property of the protein, but is a property of the entire network attached to any specific node that we're talking about, is actually pretty strong predictor. And similarly, principal component 10 and principal component 6, which have to do with which kinds of domains and motifs we used. You can also regress or think backwards to find the modifier genes using this trained machine. So we did that. And we found a lot of predictions, and these predictions are actually human proteins. And lo and behold, there is one. That was the one, only one found out by this massive, massive human genome-wide mapping, genome-wide association mapping that was found. Fanconi's anemia group 1, or FANCY, I call it. It has something to do with DNA mismatch repair. And people published it and were scratching their head about 7 years ago. They didn't know why a DNA repair gene would be important as a genetic modifier of Huntington's disease outcome. But we found this as a strong predictor from our machine learning algorithms, including many of these which happen to have both protein folding association and DNA repair association. So we went ahead and, yeah, so that's what it says, the FANCY. So we went ahead and expressed the mutant Huntington's disease protein in Drosophila eye, and the eye gets bad, like here. And then in addition, put either siRNA or overexpress some of these predicted human genes, orthologs in Drosophila. So these are Drosophila gene counterparts of the human genes, and we can make them better. Not in this particular case, but in these cases. So in fact, it is working. And we go to C. elegans, which is a very nice model of brain for an invertebrate, exactly 302 neurons with my collaborator Paul Sternberg in Caltech. And the previous one was done with Katerina van der Rohe in our institute. So we can now begin to express the human Huntington's disease protein, the mutant form and the wild type form, using a promoter that expresses it in all neurons, including the touch neuron. And we take day one young adults and light touch the tail, and the worm recoils. But if the mutant protein is expressed, then the recoiling frequency is slow or less. And then we look at this recoiling frequency in the presence of some of these candidate modifiers, which are C. elegans orthologs, not human orthologs. So human predicted, or human orthologs predicted by our models, they're orthologs in C. elegans. Do they modify or not? So yes, they do. So these are the two, these are, this one and that one are the negative and the positive controls. This is very significantly different, very significantly different, very significantly different, not different. And there are two types, the enhancers and suppressors. We can't, of course, using machine learning, tell which kind it is, but we can find both. And as in Drosophila, we also found in C. elegans. So this is a summary. A few months earlier, it's better now, but I don't have the current data formatted. And so there is some correspondence between the Drosophila and the human. And as expected, many of these are already known to function in DNA metabolism and chromosome dynamics. So what did we do? Experiments in yeast and serendipity led us to a disease concept of how molecular function works, must work in the human brain. And then the idea that networks embody information, and then learn the hidden knowledge in a large data set, and who knows how it learned, because that's what the gradient boosting model works. Then evaluated whether the machines are learning well, and used fruit fly and C. elegans, the worm, to test those models. And that's where we are. The yeast work was done by Biranchi, who started this whole thing in my lab several years ago. Sonali, the PhD student, who actually graduated last year, she did all of the computational work. Katerina's lab and Radek did the Drosophila. Songjun Koo is a scientist, is a chief scientist in Jetty Power, actually. He's the machine learning specialist with whom we collaborated, and he was a joint advisor of Sonali. And Paul Sternberg's lab went the PhD student who worked to find the worm homolog. Thank you very much, and I'll take questions. Sorry about going over time. Hi, how are you, Alka?
How are you, Animesh?
Very good, very good. Good to see you.
Lovely talk, wonderful talk.
Thank you.
It seems like a lot of I mean, I'm so glad that you're identifying the recombination and chromosome dynamics.
We always meet at the same place we began, didn't
Right.
we?
Because it makes beautiful sense with respect to the triplet repeats.
Yes.
Right? And
yes.
there's I know there's a huge body of work that's been done in yeast among many on triplet repeat diseases, triplet
Yes.
repeat instability stuff. So is it essentially all of those, or are there ones that are specifically expressed, specific systems expressed in neuronal cells? And I have a follow-up question.
Yeah. Okay, so let me try to understand your question. So what you said is that in yeast, there are triplet repeat instability genes, and mostly these are DNA repair recombination genes.
Many of them are.
Many of them, mostly. And the question is whether these genes are expressed in the neuronal cells.
Well, there's so yeast don't have neurons.
Yeah, of course.
Confused you. But a lot of these, of course, a lot of these yeast genes have homologs in mice and humans. Right. And so my question was, do these homologs, are there among these homologs some that are specific to
specifically express? I see. I did not look at that, but my guess is that most of the DNA repair recombination genes that have obvious association with repeat instability, they are really expressed in nearly all cells. For example, the MSH, MLH types of genes, they're expressed in all the cells. Yeah. And believe it or not, one of the significant signals, in fact, other than the Fanconi's anemia group 1, the FANC-I gene, the second most strong signal, it did not reach threshold cutoff after multiple hypothesis correction in the genome-wide association studies, was MSH6.
Okay, which again makes sense because mismatch
exactly.
Effects. So my follow-up question is, did you look or did you find mutations in genes involved in transcription-coupled repair? Because with a gene that's so long, transcription-coupled repair, I bet has, I don't know whether it has a big effect, but I would guess that it would have some sort of effect.
Right. Yes, we found it in yeast as a suppressor when we overexpress it. One of the rads, I forget which one. I forget which one.
There's a whole bunch of
yeah.
Rad 54 or something?
Yeah, Rad 54, so far as I recall, Rad 54. However, that did not turn up in the human. Doesn't mean that it doesn't work, but it didn't turn up, and so we haven't tested.
Yeah. I mean, one of the problems that makes this approach, it's easier to identify them in yeast, but the higher you go in terms of complex eukaryote, the more redundancy of function.
Right.
So
yes,
yeah.
yes.
Anyway, thank you.
Sure. Any other question?
Yeah, I have a question.
Hi.
You can see me.
Yes.
Okay.
Hi, Sandy.
Yes. Just wondering, when you're testing the genes you define by machine learning, are you overexpressing or knocking out, or both?
In yeast, we in our lab only did overexpression, but in Drosophila and C. elegans, we are doing, depending on what construct we have, we are doing both or one of the other, or one of the two. So in fly, we are trying to do both as far as possible because we have the wherewithal to do it quickly. The overexpression is on the basis of a GAL-induced, synthetic promoter-induced overexpression, UAS, GAL-UAS. And the inhibition is because of a siRNA, again, induced by the US, gal-US.
Right.
In C. elegans, it's a gal synthetic promoter, which is cold sensitive gal. They made it work very well. That drives a siRNA. We have not done the overexpression because we didn't get around to doing it.
Right.
We want to do CRISPR mediated knockout because that we have got going, but we haven't done it yet. So overexpression has not been done in the C. elegans, but only siRNA.
But I assume you're doing it in the wild type as well as in the
Yes,
Huntington's model, because
of course, yes.
knocking,
Yeah, absolutely. So let me share the screen again. This S is the percentage response minus the baseline divided by the mean baseline. This baseline is determined by the wild type with the mutant protein.
But in terms of therapeutics, obviously, knocking out or overexpressing a gene may have a severe deleterious effect.
Yeah.
As well.
Yeah. This is merely gene identification. I haven't got a clue how to go from here to the clinic.
Okay. Yeah. Good. Well, thank you.
Thanks.
I have a math question, actually, if nobody else is asking anything.
Then I'm not the one to answer, but go ahead and ask it.
Well, I guess, no, it's a very simple question because it's a biologist asking this. Do you have a good feel for why the SVMs and the random forests didn't do very well?
Okay, so that goes down to the fundamentals of the math or the computer science, whichever we call it, of machine learning. And I'm not the right person to answer that. And I'm sure that there are people here in the institute who are better able, and I'd be glad if somebody takes it over. Anybody?
Peter is out of town, and
I see.
Yeah.
Yeah.
Well, one thing is that it appeared that you had a lot of different variables compared to the number of samples,
Yes.
right?
Features.
And,
Yes.
right. So that's going to make it very difficult to identify things. Right?
I see.
Yeah.
Mm-hmm. Yeah, the training set is very small compared to the total. Yeah.
Cut down the features.
Yeah.
So one way to deal with that is, as you were trying to do, find ways of cutting down on the number of features, combining them, or finding some way of reducing that dimensionality.
Yeah. So that's why I suggested doing PCA, and PCA did something, but my guess is that that's not the most optimal way of doing it. Some other dimensionality reduction technique might be better. I'm sorry, I couldn't hear.
I was just going to ask very quickly if there was any way to do causal reasoning on the network itself rather than actually going after some abstraction which is not really very interpretable by a human.
Yeah, that's a great question. So causal network modeling is something that is definitely doable, and I would love to think about that, but so far we haven't. Yeah, that's a great point, and I think it should be done.
Any other questions? Anka, it's okay to ask more questions.
Maybe I'll ask a question
students?
about maybe I'll ask a question generally about the work that you're doing. So you've kind of given us this vision of maybe how to make some impact on Huntington's disease, and that's really excellent. Do you have any kind of feeling as to kind of how broadly applicable these techniques might be for kind of highly complex diseases like kind of obesity as opposed to very simple Mendelian systems?
Right. My guess is that it's going to be very difficult to do. So, and the reason is, in some cases where the genetic component is strong, such as in type 1 diabetes with specific mutations, or in genetically determined predisposing, specific predisposing mutation cases for obesity, and I think there are some, maybe it is possible, because what one needs is a handle on few specific genes or proteins with strong effect. Other than that, my best guess would be that it's going to be very difficult because you have then a many-to-many mapping, many genes interacting with many other genes, and that probably will become intractable quickly. On the other hand, these days, deep learning and all that may be able to sort through some of these. That's my instinctive response, but I don't have a better feel.
Yeah, thank you.
Okay, on that note, I want to thank you, Animesh, for a very enjoyable and interesting talk. And unfortunately, we're not gonna be able to take you out to Eureka, so we're gonna have to make that happen.
Raycheck.
After we exactly.
Thank you very much. I really appreciate that, and
Okay.
and it was a pleasure to see you all, and we'll get together again sometime soon. Thank you.
Okay.
Here's the after one talk. Here's the
Oh, there you go.
after post.
It's not the same.
Live vicariously through me. Sorry about that.
Thank you.
Okay, bye-bye, everybody.
Yes, stay safe.
Thank
Bye.
you so much.
Bye-bye.